Good afternoon, everyone. Uh, my name is Praveen Vemula. I'm a faculty and Dean of Research at the Institute for Stem Cell Science and Regenerative Medicine. This is a DBT institutes. We are also located in this BLIX, where along with NCBS and CCAMP and TICS. Um, Today, I'm going to talk about how we can do very effective translational research being in academia in India. So just to give a trajectory, my journey, I did my PhD from University of Science, Bangalore, where by training I'm an organic chemist. So during my PhD, I primarily interested in studying how we can design small molecules, how we can understand the self-assembly process of these molecules to generate nanomaterials. And subsequently, I went to and did my a couple of years postdoc in City University of New York, where I was primarily focused on understanding the how we can generate organic inorganic combinated hybrid materials. And subsequently, I moved to Boston, where I was at Harvard for 5 years. During there, I started exploring how we can design biomaterials for the medical applications. And from there, actually, I found my one of my first startup company in Paris, and we launched several dermatology based product there. And from there, then I wanted to have to make a decision whether I had to go to industry completely, or being an academia, I want to develop more technologies. Later, favored, so I moved to India, where I joined in STEM in 2013. I started my independent group there, and since then, last 13 years, we have been working on various technology based stuff. What I do is uh, today, in the first half, I'm going to talk about the work what we have done, some of the selective examples. I will run through it, and the second half, I'm going to talk about what are the key factors that enable us to do what we are doing. So when wanted to start my lab in STEM, one of primary focus is clinical translational research. So by thinking through, I wanted to have a clear picture. So just to depict that, we made this painting to understand, what lab focus is going to be. Okay, so this is could be Vemula lab, but it's a valley of death that exists in the clinical translational research. So our idea is using chemistry and biology, use the deep science and discovery based research you do, and develop the technology that could be used for human health and veterinary applications and agriculture sciences. That's our overall vision what we wanted to have. But that also shows that we are completely you know uh, area agnostic or disease agnostic. It gives much wider field to play around. But apart from developing technology, we are also equally interested to take them, convert them into viable products to reach to the market or the clinic. So for that, we have to practice science entrepreneurship. So we are equally interested to practice that. So with this vision, then I moved to at InSTEM. We started establishing our lab, where two major programs we run in the biomaterials and chemical biology programs. The biomaterials, where we deliver or develop the delivery systems to deliver the known drugs in an innovative way to eliminate whatever the side effects they have. But whereas the chemical biology program is a novel drug discovery program. And again, you can have these two types of technologies where you can use them as a therapeutic technology to treat the diseases, or you can use them as a prophylactic technology to prevent the diseases to occur. In the drug discovery, again, you can have two approaches where you can use this small molecular classical medicinal chemistry based approach to develop small molecule as a drugs, or you can develop novel biologics as the drug. So all our work falls into one of these categories. I'll quickly run through some of the examples what we have done. For example, in therapeutic technologies, one of the major concept what we're trying to develop is disease responsive drug delivery systems. These are highly important because when you have the fluctuated diseases inflammatory disorders, where you need to have the drug delivery system where the disease itself can decide when to release the drug and how much to release the drug. In that way, we will have much more effective treatment outcome. So how we do is, first you have to identify a biomarker that is produced only when the disease occur. Now you design a delivery system which can be implanted in your body, but in the absence of this biomarker doesn't release the drug, but when biomarker comes in contact, that should triggers the release of the drug, and your released drug goes and alleviate the disease, that stop production of your biomarker, that stop the release of the drug. In that way, your disease is controlling when to release the drug and how much to release the drug, right? But the key would be how we can design such material, right? So we are particularly focused on designing these small molecular amplifiers. You put the structures in such a way that they can undergo a robust self-assembly process to develop these nanomaterials, and which can be encapsulated, drugs can be encapsulated, keep them as a depot. But your structure is designed in such a way that when it comes in contact with the biomarker, that biomarker should be able to chemically degrade them, so that in an on-demand manner it can release the drug, right? So that's the key. I'll show you quickly like how this entire platform could be used for multiple medical applications. For example, like we developed this immune response hydrogel systems, which could be used for preventing the, you know, rejection episodes in the transplanted organs. Because, as you know, when you undergo organ transplantation, people have to take immunosuppressants for lifelong, but that causes their entire systemic global immunosuppression. These patients will be highly vulnerable for a small infection, could be lethal for them. And also like when they get exposed systemically, like it causes severe other non-specific toxicity. For example, like almost every hand transplantation patient within 3 years they comes back with a renal failure because of the high systemic exposure. So we have developed this, for example, like injectable hydrogels where you can take immunosuppressants post transplantation. We can keep under the skin subcutaneously depot. They sense the immune activation and release the drug only when it's needed, and it's much more localized manner. So because of that, like what we have shown is like once in a 2 months, one injection is sufficient to protect the graft from rejection episodes, but without causing any global immunosuppression, all the other systemic side effects. And as long as the animal is alive, you can protect the graft from rejection. That we have demonstrated in small animals like rats and also in the pigs. Now studies are going in the monkeys. Once that is completed, we are planning to go for the human trials. In another scenario, this inflammation responsive material could be used for effective treatment for the arthritis, because primarily it affects all your joints, right? But people have to take systemically much larger doses that causes severe side effects. So to eliminate that, people started doing the local injections in the intraarticularly. But yes, today's biomaterial, whether irrespective of you have inflammation or not, they release the drug sustained manner, few days to few weeks and disappear. But according to clinical protocols, you can do only 2 injections per year. Otherwise, you end up damaging the joints by injections, right? So what we have shown is like this disease responsive biomaterials. If you inject them intraarticularly, if there is no inflammation, they don't release the drug, despite of all the mechanical stress they undergo by walking and all. But like whenever inflammation flare comes, that triggers the releasing on-demand manner and reduces inflammation. So in that way, like significantly frequency of injections can be reduced. And in a third example, like we have added not only inflammation responsive but inflammation targeting design. Particularly like when you're talking about colitis and Crohn's diseases, right? You have patches of inflammation throughout your gut. But today, if you take any oral drugs, like whatever the dose goes to your inflamed tissue, your neighbor healthy tissue also get the same dose. That causes severe side effects. Multiple drugs like Vioxx and Bextra was called off from the market because of that. So what we have done is like we developed this orally ingestible nanoparticle systems, and which is stable in your entire GI tract, but it sends your gut. But wherever you have the healthy tissue, they don't interact. But wherever the inflamed tissue is there, like a glue, they go and bind to only there and released in a localized manner. In that way, like we can completely eliminate or like minimize the exposure to your healthy tissue. So again, to translate all these technologies, we formed a startup company in Boston. And for one of the clinical indications, we have already completed phase 1 clinical trials. Now we just made an agreement, one of the big pharma, to conduct the phase 2 clinical trials, and we are working with FDA to take that forward. So I'll take like a couple of examples in prophylactic technologies. In this case, like we are particularly interested in developing technology to protect farmers from pesticide-induced toxicity, because pesticides are neurotoxins, right? They are designed to be toxic. They're designed to kill the pests. Typically, what happens, you have this enzyme called acetylcholinesterase, which plays major role in all your neuromuscular functions by controlling the concentration of acetylcholine at the synapse and neuromuscular junctions. But all these pesticides, they are designed to go and covalently attach to the enzyme, and they inhibit irreversible, they inhibit them, right? So when you have the quantitative inhibition of this enzyme, that leads to severe neurotoxicity, paralysis and death. That's how all the pests, they die when you spray on them. But unfortunately, they don't discriminate between pest and human, right? Because we have same enzyme, same function. So when you get exposed, we have the similar toxicity. And this is the typically farmers in India and developing country, they spray pesticide without having any protection. It falls on their skin, they inhale. It's almost like walking under the poisonous rain, right? And when they get exposed, like they have not only chronic toxicity, but acute toxicity is very severe. And they have like tremors, like vomitings, and fevers and breathing disorders. If the eye doses can lead to loss of vision and death and so on. And this happens every season across the country. So we thought, like, can we develop some of the protective technology to prevent the exposure to these pesticides? So intuitively, the first one what we thought is to develop a dermal cream which can they apply before they spray, which can prevent the exposure. But like now, this cream shouldn't be like in a physical barrier gel, because these are the compounds designed to penetrate cuticles itself. No physical barrier would prevent them. So that's where like we have designed a chemically reactive gel. It's like a chemical scissor. What it should do is like sit on the skin, and if any pesticide comes in contact, chemically it should react with it and deactivate, so that it can no longer inhibit the enzyme. In that way we can prevent the exposure. And we have done extensive studies to show that such gel can completely prevent the exposure and reduce all the neurotoxicity that's associated with the pesticides and so on. But although like we worked very well this technology, but like we are still skeptical, because if you think from the user point of view, like there are two problems glaring to us, right? One is like the cost, because since it's a cream, every time they spray they have to apply, so that means they have to repeatedly buy. And farmers, their purchase power itself is very low, so it wouldn't work. And the second is the compliance, because it's not just open places, right? They have to apply all over the body every single time they spray. So everybody might not be comfortable, so the compliance will go. So that's where we thought, like, can we develop much more user friendly technology which they can easily adopt. So that's where like we started looking at like what are the practices they have. Like typically they use like regular clothes. Sometimes they think that clothes can prevent, and they wear like full sleeves and wrap the towels around and spray. But unfortunately, no cloth can prevent the exposure. And but what we thought is like, imagine take the normal cloth but convert them into an active fabric, which again, like gel, if this can also deactivate the pesticides, we can able to solve the problem. So again, like we designed a particular chemical scissors which can covalently attach to the fabric. Now this activated fabric can stitch into a gown and a face mask and so on. And again, we've done extensive studies to show that like when you have this active fabric, it can completely prevent pesticide toxicity and lethality and so on. And this is highly washable, reusable fabric. Again, to translate this technology, we formed a startup company in Bangalore. And this is our final dress, like we call Kisan Kavach. It comes with like a pant and shirt with a full hoodie and the inbuilt mask, so that we can comprehensively prevent the exposure. And this has been launched like last year, a year back. But like since then, like across the country, almost in 8 states, for thousands of farmers started using. And although like it works very well, we are skeptical, whether how the adoption is going to be. But to be surprised, like we have seen like 100% complaints so far, because every single farmer, when they spray, even one single time they use it, second time they are not spraying without using that, because they see the instantaneous relief completely. So now we are working with multiple, like state governments and other pesticide companies and all, to see how we can take to large number of farmers across the country. Okay. So with that, I'll take like one more example, like where we have been developing novel blood bags to improve the quality and the shelf life of the stored blood. Because typically, if you see, healthy blood transfusion is a life-saving process, right? In India alone, if you see, like we need about like a 12 million units of the blood for transfusion. But like through like blood banks and blood donation, you get about like 10 million units. But despite of this scarcity, every year, like almost 5 liters of the blood, 5 lakh liters of the blood is completely discarded. So the reason is, when you store them, like the quality declines. So typically human blood you can store for 42 days, but although you can store for 42 days, only up to 21 days stored blood is safe to transfuse, because after that your quality declines. The reason is, like what happens, they start developing like producing these dams, like a damage associated molecular patterns. These are like extracellular DNA, nucleosomes, polyunsaturated fatty acids and so on. All of them, they start attacking RBCs and cause severe oxidative stress on that. That's how your quality of RBCs goes down. And when dams containing blood, if you transfuse them, that can cause severe systemic inflammation, and that leads to multiple organ failure and so on. So that's why people have been trying to develop the technology to improve the quality. So, but so far all the approaches were developing various preservative solutions, because all these dams causes oxidative stress on RBC. So these are the solutions, nothing but they are buffers, right, to prevent the oxidative stress on RBCs. But the overall output were rather moderate, because one of the major factor was ignored is your damage causing agents are still present there. So it's always a war between your dams and your preservative solutions. So what we thought is like, instead of that, if we can scavenge selectively all your dams as they form from there, then can we able to prevent the loss of RBC quality. But if you think they are very diverse structures, right, how we can develop a single material that can scavenge all of them. But if you think from the chemistry point of view, they all of them are charged molecule. Either they are cationic or positive anionic charges. So we have designed the polymers which have the complementary charges and also DNA intercalating agents and so on. We can convert these polymers into nanofibrous scaffolds, which can use them as a bag. Just to summarize, you can take this dam containing blood, and if you pass through these bags like even 5 minutes, that can completely scavenge all the dams without causing any RBCs loss and so on. And just to summarize, like when you use this technology, even 42 days old stored blood quality is as good as freshly collected blood that you have shown with the in vivo blood transfusion tests and so on. And not only that, you can push the shelf life as well up to 25%. That means like not only 21 days stored blood, but you can 51 days stored blood you can safely transfuse by using this technology. Again, to translate this technology, we formed a startup company in Bangalore. Now we are working with the various blood banks and clinics to see what are the storage condition they have, what would be the design for your final device, whether bag form is better or the simple insertion would be better, because we want to get the feedback from them, which they can easily adopt it into their supply chain. So once it's done, then we'll go for the human trials. So with that, like I've shown that like how systematically we can use biomaterial based approach to develop the technology which can treat the diseases and prevent the diseases to occur. And also like we also develop various drug discovery programs. So like for example, one of the technology what we developed is small molecules to regenerate the gut, because when you have the IBDs, people have this leaky gut, right, that causes continuous infection. And we develop small molecules which can activate your intestinal stem cells to regenerate the gut. It restores the barrier function that completely modifies your disease. So at least in animals we have seen completely curative phenotype. So again, like not only developing technologies, then we wanted to translate them together. So that's why we started several companies to translate them. These are the 4 companies which are commercial sales companies. We have launched various product through these companies. And we have 2 pharma companies. One is drug delivery based company, one is pharma based company, like they are clinical stage companies now. And 2 early stage companies what we formed, they're like probably next 2 to 3 years we'll be ready to go to the clinical stage. So again, like all this work, like last 12 years what we have done at the academic setting, being there, like I could able to co-found these companies. I'm the co-founder and director of these companies. Then we have an operational team separately, we co-found with them, who runs the day-to-day operations. So uh, so then if you think back and see, like what are the key factors that are enabling us to do translational science being in academic setting, right? One is like your choice of research topic what you take. We are complete like impact centric when you pick the problems, rather than what is our comfort zone, but like we look for the problems where is a huge unmet need is there, where that you have the proper probability of developing first in class technology, then we will have the larger impact. And also pick the problems like both like local problems and global problems, because for example, if you take like uh, pesticide technology and all, like it's completely affected in Indian farmers, severely affected, right? But once we develop this technology now, like we are getting a lot of response from Latin American countries like Brazil and Chile, and they wanted to use this technology as well. So that's why, like if you can develop first in class technology, whether it's a irrespective of whether it's local or global, it will be adopted. And also like the choice of institute, I think like that plays like a major role. For example, when I wanted to come back to India and start the my independent research, I had this dilemma. Like I also had like a good response from where the chemistry based institutes like NCL Pune, like IICT Hyderabad and JNC. But the dilemma what I wanted to have is being in a taking a job in a chemistry centric institute or the chemistry department. I wanted to go to a completely biology based institute, where uh, because chemistry I can read and like my own, like I can get updated, but I don't think I can sit and read alone biology and learn. So but where if I surrounded myself with all the biologists, I was forced to learn. So that's where I chose to go to INSTEM. And like we have completely very vibrant research ecosystem, like including NCBS, and now TICS also have joined. And one more important factor is the CCAMP, as an incubation center, it promotes the entrepreneurship. I think that's being there, interacting with them, it's a great learning experience as well. That's why look for your institute which can support your long-term vision, long-term plans, right? And other one is collaborations. I think wherever it's possible, definitely explore the collaborations. We extensively work with the clinicians and both industry partners and so on, because the kind of clinical inputs they give is now gives decides what kind of technologies we should develop. Like we extensively work with a hand transplantation surgeon, gastroenterologists, and people who treat all the pesticide poison patients and so on. It's highly valuable to discuss with them. And also generating the research group. I think the building research group, this is again you need to customize the plan what you have. By definition, for these technologies, you need to have very multidisciplinary approach. So one is we have to collaborate, but in the is possible build in your research group itself such multidisciplinary system. Our students and postdocs, if you see, we have hardcore organic chemists needs and biotechnologies, biology background, even medical students and so on. They work with each other, they learn from each other, and they can develop these technologies efficiently. And again, about graduate student and postdocs, and these are the numbers. Typically, when you come back, like one of the biggest dilemma we have is because overseas and all, we have seen very vibrant postdoc culture, right? Like one of the doubt we have is, can we get postdocs here, how we build them? And I can tell you, like after 12 years, it is possible to build your postdoc culture, but you have to take an active effort to go and build them. And again, but it's comes back to the what is your working style, so which suits for you. So based on that, you build your group. I prefer to work like a lot more postdocs. And if you see any given point of time, either 1:2 or 1:3 student to postdoc ratio what we have. But at the same time, we also make sure that they get like what they deserve, and what are their trajectories, their plan, career plan they have, and nurture them to so that they go and pursue their dreams. So all the alumni, some of them you see, they have been pursuing their own careers. Many of them took independent faculty positions from they are in IITs and DBT institutes, NIPERs and private institutes and state universities and so on. Many of them transition into industry, and some of them formed companies. Now they completely moved to the startups and they're running their own startups. And the graduate student, they also went and exploring the postdocs. And we also have the people who took the science administration as a career. Now they are doing very well. So in that way, when you engage with them and see what are their aspirations, and support them to and help them to achieve their goals, I think in that way you can build very vibrant research culture in the lab, which will be win-win situation. So and the other important factor is funding. And all morning you have seen what all the possibilities are available there. And we are blessed with glad to be at INSEM, we get generous core funding support as well, that will enable us to go and pursue even risky ideas and generate enough data to go and attract additional fundings. So we have both national fundings DBT, DST, NRF, and Ignite Life Sciences and BIRAC funding. And also there are several bilateral fundings are available, like Indo-Swiss, Indo-US, Indo-UK, and all. You can explore depend upon where you are have the collaborators and all. And also we can also attract the international fundings DOD, NIH fundings also we can get them. And these are RO1 equivalent grants what we got multiple of them directly from depend of from defense. And also industry funding, like, again, depend upon, like, if you have very targeted manner, if you want to attract the fundings, and we have funding from both, like, large industries to small startups, both from US and India. So as earlier session they told, like, based on what is your idea, what area, then you find a right funder to then you will have to maximize your chances to get your funding. So with that, like, one more factor they want to discuss, like what we can do beyond your science, right? One thing, like, we wanted to contribute to entrepreneurship policy, because uh, when we joined in STEM, like after a few years, when we developed the technology, when we wanted to form the companies, we realized we don't have any policy framework which will enable to give the permission to faculties to go and form the companies. So much of our surprise, we figured out that none of other DBT institutes also has such ecosystem. So then what we thought, like, instead of getting disappointed, we thought of set up those policies. We work with the leadership, like, Rams and Apuva, like, we wrote down the policy framework. We also studied other CSR policies and all put together. We came up with this entire policy framework, which got approved by our general body, governing council, and which now able us to, like, easily form the companies. Now any faculty can come and take permissions within 2 weeks and go and form the companies. That's the level we set it up, and people are leveraging that. Like, now we see, like, a couple of our colleagues already started companies at InSTEM. And not only that, like, after 10 years now, when the BRIC has formed, all the DBT institutes came together. Now the BRIC has adopted the same policy framework. Now it has uniformly approved. Now every single DBT institute across the country use the same policy framework. Now anyone can form the companies. So in that way, like, we can contribute to much larger ecosystem. And another one is, like, I wanted to teach science entrepreneurship for students, because if typically, if you see science entrepreneurship, like, or like entrepreneurship treated teach most business aspects of it, but like, for the you have rarely you have any courses for the academicians who want to pursue science entrepreneurship. That's a course we wanted to develop. And thank, like, I should thank INSEM, because 10 years I didn't teach a single course there. They are generous enough to let me go without teaching, because the problem was, like, I didn't want to teach another chemistry course or biology course. I wanted to develop this course and teach science entrepreneurship. But entrepreneurship is tricky subject, like, without doing, if you teach, it's like a preaching, it's not teaching. So we wanted to see practically what things work, what don't work in our Indian ecosystem, and put that all together. So that's why, like, we waited for 10 years. After going through some of these examples, now we formulated the course. And now all the DBT Institute PhD student, they have a common course. They come and do one semester in Delhi. So that's where I go and teach this translational course to them. So with that, I'd like to stop here. And about future journey, if you see all our technologies so far, they are human centric. Um, uh, Now, but very recently we started some of the programs that can help to, like, in veterinary science and agriculture science. Maybe next 3 to 5 years they will be mature to bring them. So with that, I would like to stop here. And some of the key takeaways is basically three impactant factors. What I believe, like, it's the colleagues and collaborations, impactful problems that you select to solve them. Because when you talk about the colleagues, these are your younger colleagues of students and your peers who can have an enormous impact on the way you work. And also collaborations, as I said, like, seek the collaborations as much as possible to get both from clinicians and academia and industry, and find the impactful problems which deserve to be solved, irrespective of their local or global. So with that, I would like to stop here and happy to take any questions if may have.
We take a
Thank
couple of quick questions, please.
you.
Hi, excellent talk. So I was wondering about you. You mentioned people who are at InSTEM can start a company, but how about people who are coming as new faculty and they already have a company? So how does the time is divided, or like is it like a part of the conversation during the hiring, or is it like something that's already in your policy that you developed?
You can have that dialogue with them, and typically you are still allowed to carry out whatever the companies already have, but you should disclose them. So one of the key factors for all your entrepreneurship activities, less than 20% time you can maximum allocate it. So you disclose them and discuss with the conflict of interest whether whatever the company activities with the program, what you're going to build in a lab, what is the conflict of interest you have. You disclose all of them, it can be given permissions.
Thank you. That was a great primer on the translational landscape in India. My question is actually more about um, I- what about the bureaucratic and regulatory policies around actually taking it to the translational level with clinical trials, um, with pharmacotherapeutic agents, especially. And I understand there have been change in rules to make it more flexible with the NDCD in the recent past. But how does the registry work? What has your experience been? Just yeah.
So yeah, this policy again, regulatory body is also evolving. Right, maybe our next speakers would give us a little more detail during the panel discussion, probably. But they are evolving much better, I would say. Like it is possible. Again, like might have to go with the case specific manner. You don't get the blanket approval for anything, right? Like having a pre-meeting with them and bring them people. One of the key thing is like interact with the regulatory bodies as early as possible. Don't wait till your last stage to engage with them. So because these are directly can interact with CDSCO, but if not, there are also many people who worked with the CDSCO, have the expertise, have informal mentors. You talk to them so that like, they can give you like a what are the very critical data set that need to be generated in order to get your approval smoothly. I think that is possible, definitely. Yeah.